Okay, so my previous video was about um, carbohydrates. So now let's take a look at proteins. Protein is our second organic compound. Remember, organic is carbon based. So proteins are really, really diverse, meaning that they can be a lot of different things and they have a lot of different functions. All right. So, first of all, let's look at the functions. You can see there it does a lot of stuff. And this is actually just the tip of the iceberg for what it does. But it does things like building things like it makes up our hair, makes up our muscles. It allows chemical reactions to occur, speeds up chemical reactions. It helps defend us against microorganisms. It used for transport. And then one of the really important ones that we're going to talk about in this unit is it acts as enzymes. And oh my goodness, I should have a space there. Acts as an enzyme. So when we get to enzymes, it's important to note that proteins are enzymes. Or excuse me, enzymes are proteins. Okay. But the big thing with what we're going to do for us is to know that proteins are made of amino acids. Amino acids are the monomers of proteins. Okay, if you remember, monomers are put together to build polymers. Well, the monomer that's going to be put together for proteins are called amino acids. You are not going to be responsible for knowing all the amino acids. There's 20 of them. What you do need to know is that the amino acid is the monomer of proteins. But if you take a look down here, all amino acids are pretty much the same. They all have an amino group. That's that green part right there. They all have a carboxyl group, and they all have a central carbon and a hydrogen sticking out. Now, I'm not going to ask you to recreate these things, but important thing to remember is they all have this part here. We call it the R group, and that's where every amino acid is different. So you can see these two. I just have two up there, alanine and serine, both exactly the same up top, but then they get a little bit different down here at the bottom. That's the R group. There's 20 different of these molecules that we put together to make proteins. These 20 different amino acids can combine in thousands and thousands of different ways to make thousands and thousands of different proteins. Now, what do we call a protein? Well, it starts as a polypeptide chain. Remember, poly means many, and then this term peptide is new. We call them polypeptide chains because when we link amino acids together, they are held together by a bond called peptide bonds. So now think about this. If I have 20 different amino acids, and then these polypeptide chains are usually about 100 amino acids long, and you can put them in any different sequence that you want. Think of the millions of different combinations of proteins that you can have. Okay, so I always talk about the English alphabet. There's 26 letters in the English alphabet. Think how many words there are. There's good words, there's bad words, there's short words, there's long words. Well, same with a protein. These polypeptide chains can be a combination of 20 different amino acids in any order. And then the next thing is that it's going to be a different shape. Already talked about this. The peptide bond holds these amino acids together. That's why we call them polypeptide chains. The important thing to know is that when I build a polypeptide chain, that chain needs to be folded into a certain shape before it has its function. All right. A good analogy here is like the yarn of a sweater. If I have yarn and I just lay it all out, it just looks like a piece of yarn. But if I fold it and sew it, and I guess it's not sewing, knitting, if I knit it into a certain shape, I can make a sweater that looks like a sweater. Well, a protein, if it's just this chain laid out, it doesn't look like anything. But if I then take that chain and I fold it into the proper shape that the cell needs, then I make a protein. Denaturation, or if a protein gets denatured, it's because the protein has unraveled. Think of picking at that sweater or at that yarn. If I keep pulling at it, I'm going to pull the sweater apart. Well, that's what happens with a protein. If something happens, it's going to be due to a high temperature or possibly it's been exposed to an acid. Well, if that happens, it can cause the protein to denature, which would cause a change in the shape of it. If that's the case, it's going to lose its function. Good example there. If you ever fried an egg or saw somebody fry an egg, you have denatured all of the proteins in there. And guess what? It looks a lot different than when it came out of that eggshell. So it kind of looks like this. I have this little protein chain. This is just a very small one, right? It's going to start off by looking like that. Then we start to fold it and kind of ravel it up. And then it gets a little bit more folded up. And then finally, this is my final product. Now, so what they've done here is there's multiple polypeptides that are linked together to form the final protein. You do not need to worry about primary structure, secondary structure, tertiary structure, quaternary structure. Don't worry about that. Just think of the visual here. This down here at the bottom is what our protein looks like at the end because of how it was folded into shape. And that is all we're talking about with proteins until we get into what we're going to